I got into more. Good evening. This is Sue. I'm from New Jersey Mycological Association, and today is Taxonomy Tuesday, where members share their finds. But to start off tonight, we have Professor Kyle Root, who is going to present the results of student Megan Hartman, who received her scholarship money from us at Lockhaven University to explore the mysterious world of white nose syndrome caused by Pseudogymnoascus destructans, fungus threatening the bat populations. The chemistry-centered research delves into the complex chemical interactions between the fungus and the cohabiting bacteria, uncovering how their behavior shifts depending on glucose availability. In the presence of glucose, the microbes seem to collaborate, but in the absence, they unleash an arsenal of antifungal and antibacterial compounds turning on each other. Using GC-MS analysis and literature-based characterization, the team is uncovering molecules that may even have implications for understanding diabetes-related lesion formation in humans. With the next round of experiments underway this semester, this study promises both ecological insight and biomedical relevance. Don't miss a chance on this to hear more about these intriguing microbe dynamics and surprising parallel between hibernating bats and type 2 diabetes. Okay, Professor Root, would you tell us a little about yourself and the project?
Sure. So I'm a biochemist, a professor of biochemistry at Commonwealth University of Pennsylvania in Lockhaven. So there's three schools that are associated with the state of Pennsylvania that all fly the same overall flag of Commonwealth University. It's kind of a new thing. We underwent a university merger, but it's ultimately been a fun journey because I've met, new people and been exposed to new instruments. But I still have my old friends here at Lockhaven, like Dr. Barry Overton, who turned me on to this project, and I'll get into a little bit about what initially interested him in what I could do for the project because I'm not a mycologist. I am a biochemist. Really, I'm a biophysical chemist. I study primarily membrane proteins and enzymes. So this work is definitely not in my wheelhouse. But that's kind of the cool thing about, collaboration is people can turn you on to things that maybe you never thought you'd work on, and your strengths can play a big role in moving something forward, especially when it's at the um, boundary between disciplines, such as biology and chemistry. So Dr. Overton, he had a ton of really interesting sort of qualitative and macroscopic observations, and we're hoping to take it more into the microscopic and quantitative level, um, and really trying to understand how this Pseudogymnoascus destructans, which is the causative fungus in white nose syndrome that has been wiping out bat populations in the Northeast for the last 20 years. What makes it so deadly to bats, and what our hypothesis is it's actually when the fungus is teaming up with a bacteria. And really, to understand the mechanism behind that, we're going to have to understand what kind of molecules are at play. What protects the bat when there's only one infection of say fungus or just a bacteria, and how does the bat how does its immune system fail when these two microbes work together. So we're not talking about mushrooms really here. We're talking about fungus in the sort of microbial state. I have a list of my collaborators. Megan Hartman did a lot of the, actual science here. Jared York, I'll get into a little bit about him. He wasn't really a student of mine. He was actually a really good friend from college that got me into the GCMS. I was helping him with a master's project. Troy Dermata is our sort of he's a physical chemist here at Commonwealth University, and he was very key in putting together some of the protocols for using the GCMS, the gas chromatography-mass spectrometer system. Barry, of course, sort of, is the other primary investigator along with me. So I think that's about as much of an intro as I need to give. And I'll get right into kind of, what we're thinking about here. So, everybody knows that fungus and bacteria, they don't always get along, right? In fact, when we think about fungal bacterial interactions, you might think of something like penicillin, right? Which comes from molds and um, is really deadly to bacteria. It disrupts their ability to synthesize their cell wall. So it can break down that sort of protective layer in bacteria and allow them to more easily be cleared if you have an infection or something. So fungus, they've evolved to kick bacteria out of their local environment so that they can, of course, make the most. They're competing with bacteria for the same ecological niche. On the other hand, you can imagine a situation much like you see with many examples in nature where maybe fungus and bacteria will work together. They'll work together symbiotically, right? In a positive manner, not necessarily they're trying to, clear each other out and win some kind of war. So uh, the crazy thing about this aspect of fungal bacterial symbiosis is really there's not a lot out there on it. In fact, it was what 20 years ago, a landmark paper came out by Romano and Coulter, which was the first to sort of look at, what's going on in particular circumstances when fungus and bacteria are co-cultured. And in this case, they were really interested in in basically yeast and how they get along with certain types of bacteria, such as P. putida. And what these early research studies found is the bacteria, one of its ways of sort of trying to conquer whatever medium it's colonizing is by using a byproduct of glucose metabolism, gluconate. And basically, when this gluconate is formed through a biochemical pathway called the pentose phosphate pathway, they can take that gluconate, which I have shown up here, um, and they can either use it for other biosynthetic purposes, extract electrons from it and so on and so forth. I'll talk a little bit about that soon. Or they can export it to the surrounding medium. So, when I look at a molecule, I always kind of dissect it. And this gluconate here has this really important bonding pattern. A carbon is double bonded to an oxygen and single bonded to another oxygen. We call this a carboxylic acid. So the bacteria will export a carboxylic acid to the external medium. They'll shoot it right out of, the cell, and that acidifies the medium. And you probably realize, things you've learned throughout uh, your studies that most life likes to operate at like a pH of 7, maybe a little bit deviating from there, unless they're acidophilic, right? They can really handle acidic conditions. If they're alkaline-philic, they're going to really hate acidic conditions. So microbes can export gluconate, acting as an acid, and just kind of, acid burn everything up around to put it in sort of a simple term. So um, in the presence of glucose, bacteria import the glucose, export gluconate once energy needs are met, and that can allow them to have, a better level of proliferation. It can get rid of other fungus or bacteria that are around. Now, some fungus will actually be able to use gluconate. So they'll see that gluconate and then they'll take it back up within them. They'll import it. And by doing so, they will spare whatever medium these two things are growing in from becoming too acidic. And this can be, beneficial for the fungus. It's getting some energy in the form of gluconate. There's still a lot of energy in this molecule that the fungus can use. And really, it can sort of balance the pH change such that that bacteria doesn't overcook itself. So this particular data figure here is looking at sort of when you grow a bacteria alone in a high percentage of glucose, its growth will drop off significantly. So, colony forming units per milliliter goes up initially and drops off, and that bacteria has killed itself. It's more pronounced at higher glucose concentrations than lower. But if you co-culture a yeast with this, look what happens. You can see here with the dashed lines that the bacteria continues to become, more able to form colony forming units. So that's sort of the basic science of what's going on and what we think might be going on when PD is in a bat wing and it comes into contact with a bacteria that's beneficial to it. They may be undergoing this sort of exchange. This particular hypothesis comes from, like I said, earlier, sort of macroscopic observations that Dr. Overton has looked at, where he found that basically, the PD fungus, it's not really able to grow nicely on glucose-rich plates. If you have an agar nutrient medium that has a lot of protein, which is, peptone, something like that, uh, well, the PD will grow quite nicely. The same thing is true with the bacteria that Dr. Overton has been co-culturing with his PD, B. agrestis. It also does not grow nicely on glucose, and it's probably that it over-acidifies things. In the case of the PD, it doesn't grow great because it just needs a little bit more. It needs that protein, right? But in the case of the bacteria, which can literally make almost everything out of glucose, it's less complicated organism. It's probably acidifying the medium and growing much more poorly. So when we put these two guys together, you can see that they are able to grow on these plates quite nicely without any, sugar source. As you might imagine, it's the growth isn't great. Up to 1%, we start seeing the lush growth that we actually see. It's very similar to what we see with PD alone on peptone. So there's something going on here. And there's a limit of returns. Once you get up to 5%, it becomes much harder for the fungus to control the acidity of the environment. And these particular plates, they all have an indicator in them that goes from sort of this deep red to yellow as the level of acidity increases. So you can see that at least at 1% dextrose, the acidity is really well controlled. At 5%, maybe not so much. So there's kind of a sweet spot there. And according to Dr. Overton's work, Barry is he's seen that in these lesions and bat wings, you don't just see PD alone. You oftentimes see a bacteria along with it, and this is sort of telling us that the lesioning process is being enhanced by this sort of symbiosis. These microorganisms are able to live on the bat wing much more fruitfully when both are around, and when you have damage to the bat wing, that's when they die. I talked a little bit about jobs I've had in the past before people kind of came in. I actually worked on white nose syndrome in a different capacity when my wife was in graduate school. She was monitoring sonar of bats in the Crystal Lake region of Pennsylvania. And I used to help her carry car batteries out that would power the the sound collection devices. So it's funny to kind of come back around and look at this in a different way. But carrying car batteries in the summer was really not super fun. But then again, she was happy that I did it for her. So going back to what we're actually here to talk about, this observation that there's higher virulence of PD when we have some bacteria around compared to PD alone leads us to, trying to have some understanding of what's going on. And we're using that prior work to inform us. We think that the gluconate production, that gluconate going into the medium, gives PD an additional source of carbon. It allows the bacteria to survive in the medium longer before it becomes too acidic, even for itself. Interestingly enough, diabetic bats are less prone to, these to bacterial infections in general, and also infections by PD. So this is sort of where that connection to diabetes comes in that this is very counterintuitive that high blood sugar, we think of diabetes in humans, right, as you might need to have a amputation done because you get some kind of chronic infection due to lesioning and things like that, maybe in your legs. But diabetes is actually protective in bats, and it's because it causes the bacteria, which can cause the lesioning, to burn out too soon. So when you have PD as a sort of a bank for that acidic molecule to go to, you circumvent the glucose toxicity. So the bat's diabetes is no longer being advantageous in helping them resist wing infection. So, ultimately we want to understand this interplay, but we think that there's more than just this story about gluconate exchange between the bacteria and the fungus. I mentioned, for instance, the fungus loves to grow on protein. So another hypothesis I'm not really presenting here is we think there may be some exchange of nitrogen between the bacteria and the fungus to allow the lush growth. And there also may just be other metabolic changes going on where you don't have to monitor what's going on with the gluconate, for instance, that may tell us, symbiotically, how are these two organisms, keeping things well balanced where both are successful. So at this moment, I think I'll pause for, does anybody have any questions so far? Is everybody kind of following along? You can be interactive if you'd like, or I can just roll through the whole thing. But
how do bats get diabetes?
How do they get diabetes? I think it's similar to humans, right? You can just have genetic mutations that either you don't produce enough insulin or your insulin receptors are not being activated by insulin as well. So it's just genetic, right?
Is
Bats
that
have
common?
quicker population turnover. They have higher evolutionary rates. So when these pressures are put on them, a diabetic bat may be right selected for, so long as they can do everything a bat needs to do and survive. And if it confers an advantage against bacteria, then these mutations affecting your insulin signaling can be of advantage. So it's no different than humans. I mean, they're not getting it through diet issues. They're coming upon it honestly, so to speak. So that's a great question.
Yeah, genetically, as opposed to just overeating.
No, they're not hitting up the candy shop. They're not eating the Little Debbie's or anything like that. Whatever that might do.
Yeah, because they're
Um,
eating mostly protein, right? Or
yeah, a lot of protein there. So it is kind of confounding, but it has to do with just regulating your blood sugar because, um,
I
go
I
ahead.
have a comment or question. In my mind, symbiosis is just a living together, and it can be different. It can be characterized by parasitism, commensalism, and mutualism. So how would you call the association with the PD and the bacterium?
That's such a great question. I would probably, I'm a biochemist, so those terms are pretty alien to me, but I would say it's mutually beneficial, you
All
know.
right. So what you really need to say is that it's a mutualistic symbiosis.
Yeah, okay. Yeah,
Yeah,
that's a
I'm
good
sorry,
comment.
I'm a retired high school science teacher. So
No, very good. No, anything like you're right because it's not exactly clear, but certainly I don't think parasitism would be the correct one. I think it's a mutually beneficial. You scratch my back, I'll scratch yours. We don't know exactly beyond, oh, here's some carbon food and I'll lower the acidity for you, beyond that, what's going on exactly. But I think you'll find that there is a little bit of also competition going on as I present some of the other things we've seen.
and actually, how do they meet each other,
I mean,
or are
I
they
think
just around?
it's they're just around, and it's just by chance, right? And then once you have a co-infection of these two things, bats are living in clusters, so they pass the two things at once.
Right.
It's just like people. So, there's no other questions. Those were great questions, by the way. I'll go on and just talk a little bit about, what we're going to actually look for a bit to make sure that this is probably what's going on, uh, uh, at least, in part. So here I'm showing the initial steps of the pentose phosphate pathway, which probably everybody here knows glycolysis, but once glycolysis is generated enough ATP, um, what are you going to do with that excess sugar? So there's other things you can do. Glucose is phosphorylated inside cells that traps it there because there's no transporters to let phospho hexoses out of the cell. So once you import some energetic material, you don't want it to leave. It gets a little phosphate group put on it. So we start the pentose phosphate pathway with glucose 6-phosphate. And then an enzyme called glucose 6-phosphate dehydrogenase is going to oxidize glucose 6-phosphate into 6-phosphoglucolactone. All right, so we steal some electrons from this carbon that has the red bonds to the OH and H, and we end up making something called NADPH, which is a biological reductant. It's used in a lot of anabolic reactions, but that's that's maybe a little reductionist. It's an important molecule for energy exchange in organisms. Then we have something that can be processed further. An enzyme called lactinase will actually hydrolyze the 6-phosphogluconolactone into 6-phosphogluconate. And this is sort of where the organism can decide whether or not we want to export this or not. So this 6-phosphogluconase has to be dephosphorylated by an enzyme, which hypothetically, in my mind, is probably 6-phosphogluconate phosphatase. And you end up with gluconate or gluconic acid. More likely, it's gluconic acid, which is a little bit more hydrophobic than gluconate and probably gets sent through a transporter, such as this gluconate transporter. An example of which is from E. coli. So these over here are called ribbon diagrams, and these are just kind of the bones of what a protein actually looks like. How does the polymer of amino acids transmit through space? So here I've got a structure of 6-phosphogluconate phosphatase and the gluconate transporter, which I think are two important proteins that would be increased in their abundance within the bacterium if gluconate export was being sort of upregulated and turned on. These structures are actually predicted structures using a very sophisticated database, which has looked at all the sort of known, proteins and the known genomes of all the organisms that are out there. There's something called AlphaFold uses deep learning to predict protein structures, which as a graduate student, I thought would never happen, but here we are. And it's kind of cool because you don't have to necessarily solve the protein structures to look at them and understand how they work. But proteins are doing all the cool stuff with these small molecules. So we like to look for changes in their levels to know what kind of genetic level changes, are being turned on by alterations in the, environment, signals in the environment that are affecting the microbes. So once we have the sodium gluconate, for instance, or gluconic acid, gluconic acid exits the bacteria, and then it dissociates, it drops off an H plus, or it makes H3O plus, I should say, and we end up with the weak base salt of gluconic acid, sodium gluconate. Sodium gluconate is what we think then is going to be imported back into the fungus. So fungus has gluconate transporters. This is known. Once the gluconate comes in to the fungus, it also has the pentose phosphate pathway. It's a very well conserved ancient biochemical pathway in all organisms that at least I know of. And the fungus is going to want to trap that gluconate inside of itself to use for its own metabolic needs. And that's probably going to involve this gluconate kinase. So actually this structure has been solved. So some group of researchers got the crystal structure and submitted it to the protein structure database. So gluconate kinase installs a phosphoryl group at the 6th position to make 6-phosphogluconate. We got that back. A little bit of energy gets put in to do this. We burn an ATP, but ultimately you get more energy out by then oxidizing that 6-phosphogluconate down to ribulose 5-phosphate. So this oxidation involves the loss of a CO2 molecule and the production of a biochemical reductant, which is actually worth 2.5 ATPs. So you end up netting 1.5 ATPs for what it's worth. And ribulose 5-phosphate, what can a fungus do with that? It can build nucleotides. It can make more NADPH, which can ultimately help make more ATP. It can be used as a carbon skeleton for making other metabolic precursors for biosynthesis. So there's, for instance, amino acids. It can be used to help make amino acids, nucleotides, so, parts of DNA. So it's a really awesome molecule to be importing in. I can see why the fungus would like to hang out with somebody that said, hey, here you go. Here's something sweet. So anyways, I'll mention more where we're going with that stuff towards the end with the future directions. But like I said, we think that maybe there's other cool things that are changing at the level of what, biological pathways are being turned on in both these organisms when they're in contact. So one thing we can do is we can analyze, what are these things exporting? And in particular, we were interested in sort of the volatile things that they might be exporting as the volatile things. They may be a little bit hydrophobic. They may be, things that are going to be sort of conducive to the GCMS process, in that we can collect them in the headspace of our fermentations. So ultimately, how do we collect these volatile compounds that are being made under these different conditions, say, PD growing with the bacteria versus PD growing on its own versus the bacteria growing on its own? How are we going to look at what kind of molecules these things are sending out, and these molecules are escaping into the the environment? And I think if anybody's smelled a beer or something, smelled a fermentation, that there's plenty of things that are exiting that medium, but those things have important roles while they are dissolved within the medium, and as well as when they actually volatilize to attract things to maybe that space where the fermentation is going on. Anyways, we use these things called solid phase extraction cartridges. And basically, I have got a little schematic of a solid phase extraction cartridge. It has this plunger that is housed in this sort of solid, metal unit. and the plunger can be depressed. The head of the unit is actually a syringe tip, but when you depress the plunger out of that syringe tip, a little polymer coated fiber comes out, and this fiber has a sort of hydrophobic coating. You can see this coating material over here on the right side, this methylated silica oxide. So those CH3 groups make the usually polar, glassy like silica oxide non-polar, which will allow it to bind to molecules that are floating around in the headspace of some kind of growth that you're doing. So this polydimethylsiloxane that you see here is coated at the tip of this fiber at a very, it's a very thin film, 100 micrometers thick. And ultimately what we do is we push this into where we've grown our PD or our PD and bacteria in different mediums, depress the tip, and that tip just sits there in the headspace. It doesn't dip into the liquid, which is in this little brown bottle, and it sort of collects those volatiles efficiently on the surface. They absorb over time until you saturate the tip after some amount of time that needs to be optimized sort of empirically. So we just basically found that you get no changes in the signal on the GCMS after about 2 hours shaking this this whole apparatus at 150 rotations per minute and holding the temperature at 37 degrees Celsius. So that's kind of what's going on here in this live photo that I took. We're actually extracting the volatiles out of the headspace, the region above the liquid that is available from what's going on in the culture. And then we want to know what's absorbed to that syringe tip. So we use a technique called gas chromatography to separate all the different molecules that absorb to that tip by their polarity as well as their volatility. So the gas chromatography it's just a really long capillary tube basically that has been the interior has been coated with something called carbo wax, which is sort of similar to the PDMS in a way. It's a little more polar, so it's not exactly the same, but it has properties that allow small molecules to bind to it. Now, these molecules aren't going to sit there stationary as they did on the SBE cartridge, because we inject, we put our tip into an oven, and the oven increases in temperature. The more volatile something is, the sooner it gets, turned from being absorbed on the tip into the gas phase. And then we have an inert carrier gas, helium in this case, that's going to push that molecule that desorbed through the GC column. So there's kind of a lot of different elements of separation going on here. There's the polarity from the carbo wax that allows molecules to be separated, and then there's their different volatilities. So by using a really, 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 really long, like these things are many meters long typically, column, we can separate hundreds of different analytes from each other. So you can get a really complex sample and know absolutely within some level of certainty, what that might be. How do we know what it might be? Well, we've separated all the molecules, and then they go from the GC end of the system to the mass spectrometer end of the system. And the mass spectrometer is just going to take those molecules and it blows them apart using an ion source. It puts charges on the different fragments, positive, negative charges. And then these fragments go through a series of magnets that deflect them based on their mass to charge ratio. So something that has a really big mass will, it's not going to be deflected very easily compared to something that has a small mass. Something that has a high charge is going to be deflected more easily than something that has a low charge. That's sort of how you might imagine the physics of a magnetic separation working on charged species. Depending on those mass charge ratios, these molecule fragments hit detectors at different places. Only an individual, species hits the ion source at one time because they're all separated. So this stuff's all time resolved. and then the fragments get separated from each other, and then we have a computer, basically look at all these different fragments that we've detected, and it compares it to a library of different knowns that have known fragmentation patterns. These fragmentation patterns follow a chemistry of, the logic of chemistry. So it's like a fingerprint almost. And we can look at this fingerprint database and we can say, oh, well, that is ethanol coming off there based on that fragmentation pattern. So human beings can sit there and they can look at the fragments themselves and they can put together an idea of what that molecule might be. But nowadays we just use these library searches, and we get pretty high confidence for a lot of the hits we get at this point. So how did I get into this particular work? How did Dr. Overton and I start collaborating? Well, actually, I was working on something totally different with this master's student, a friend of mine from undergraduate, Jared York. He was interested in actually brewing using, instead of wheat as a carbon source, which the price of wheat is really fluctuated due to the war in Ukraine, and the price of wheat is kind of a little bit, in a sense, high, given that you have only certain seasons you can grow wheat in. Wheat is growing in a two-dimensional space. He had the idea, well, what if we use mushrooms? Mushrooms, right? They can grow in stacks. They could grow all year round. And because they have that cell wall, they can give you carbs for fermentation. So he was just trying to, make some interesting cold ones. And what we did was we used this SPE cartridge paradigm to look at, hey, how does, relative to a brew using wheat, how does changing out that wheat using oyster mushrooms or button mushrooms or shiitake mushrooms, how does that change the chemical composition, of that beer? Because what's coming off it is what you taste, right? Scent is a lot of what taste is. He wanted to make sure he wasn't going to send something nasty out there, or just, have a really good idea of, can I make the same beers that I've developed over the years using a totally new carbon source? So here's an example GCMS chromatogram. All these peaks are individual molecules. The intensity just kind of a measure of how much, but this isn't really being done in a quantitative way, so take that with a grain of salt. And you can see that the different peaks come off at different times, which is manifested by the fact that they're being separated by the GC column. So we have all these different peaks. What are these molecules? Well, I mean, there's all kinds of cool things that were showing up. Linalool, ethyl caprate, citronella acetate, phenylethyl alcohol. I mean, it's a whole battery of different things. Geranol. Geranol is what the smell of geraniums is from. You see all kind, and it's also anti-insect. So you see these kind of cool things showing up. In some cases, you see it in the control, and you see it in all the different brews he's done with the different mushrooms. In other cases, you may not see. There's completely unique compounds that are showing up only in the certain context of what mushroom or lack of mushroom or percentage of mushroom that you're putting in there. So we were able to see a lot of very interesting things going on. A lot of different esters, such as ethyl 9 decanoate. And I had the question, why are they making these esters? Esters are really good smelling molecules to humans. And the answer is that the esters, those scent molecules, are attracting bugs. And the bugs come, they go into some fermenting thing out in the wild, they pick up the microbes, and then they go to the next spot and they deposit those microbes. So it's no different than, geese moving around fish eggs or something like that. So how do fish get into high mountain lakes? Well, it's birds. So we can look at these different compounds that showed up, and something, these things are all known compounds, and we can see how they were affecting his brews, such as, caprylic acid has a non-pleasant, fatty, oily, rancid smell. You don't want a lot of that in your beer. Tributyrin, I was like, oh, buddy, you got tributyrin in there? That's baby diaper. That's some really nasty stuff. So it's a whole world of molecules that are out there to explore. So I was just kind of talking to Dr. Overton. I'm like, I'm doing this really cool stuff. It's kind of tangentially related to fungus. What do you think of that? And he's like, I think you should get in on this PD agrestis stuff. So let's see what's really going on. Let's see if there's any of these metabolic changes, any of these unique compounds arising in the case of co-culture on peptone, co-culture on glucose, no co-culture, whatever matrix of different sort of things you want to think about. So here's a GCMS we collected. 45-day cultures grown in 1% peptone. So this is a medium that really the fungus would be happy on growing alone. The bacteria would be happy on growing on alone. And ultimately, we knew we were going to see a lot of stuff on it because they did enjoy growing on it. So that was kind of, the glucose spectra were a lot less interesting than this. Really, these library searches, they're only so good. How close is that fingerprint to this fingerprint? And we were rigorous in the sense that we said, only if there's an 85% match will we trust that this is the molecule that computer is telling us. So even though there's a lot of peaks here, really you need a lot of signal to make confident predictions in GCMS. And only 8 of these peaks were able to be characterized, in my mind, semi-definitively. Some interesting things that came up in this particular condition with bacteria only, okay, is what you're seeing here, is 2-tridecanone and 4-cyclohexyl 2-butanone. So we see some kind of cool ketone molecules appearing that have some relevance to the bacteria, and they would be absolutely volatile. So it was no surprise to find them in the headspace. Now, when we co-cultured PD and bacteria in 1% peptone broth, we wanted to see, is there anything new showing up? On the last side, you saw that there was the 2-tridecanone. So there's some things that are maintained, and you might expect this is from the bacteria, right? So that's another thing of running these single, running the non-co-cultures, that you are going to make sure that you are seeing things that, are unique when you grow them together. If we grew them together and not alone, the 2-tridecanone, I wouldn't know who that came from. One thing we saw that was unique was something like octanoic acid. We had 17 peaks that gave us the ability to assign things. And here are some of those peaks that, showed up in bacteria only. The 2,5-dimethylpyrazine, which is shown here, is an antimicrobial. So that's one way, in addition to acidification, that the bacteria may be controlling its space. And it also made an antifungal on its own. So who knows, maybe there's, of course, spores in the air that it's trying to ensure that those don't come down and colonize where it's at. In co-culture, we saw something like carbone. Carbone is shown here. It's actually got like I believe a scent similar to spearmint, which was interesting to see. It's antimicrobial. We see an antifungal. We see things involved with decreasing virulence. We see antimicrobials, antifungals, antimicrobials. Some things we don't know what their purpose is. But I think what was really amazing about this, I'm not really talking much about the pentose phosphate pathway. Those molecules in that pathway, they're not really volatile, so you're not going to see them in the headspace. But what the metadata seems to suggest here, right, is that not only is this a cool way of seeing what kind of metabolic changes happen when there's some kind of, sharing of space between 2 microbes, maybe it's a way of exploring the frontier of antimicrobials. We don't know. Maybe this 2-isopropyl 1,3-propane diol is a totally new antifungal agent, right? So you can imagine this work blossoming into the sort of biomedical realm of biochemistry and biology. So that's sort of where the story ends. Ultimately, we didn't get to analyze what was going on with the glucose very finely because our SPE fibers, which should be resistant to the temperature of the GC port, absolutely melted. It was really sad. Those those SBE fibers were a gift from my friend Jared. After he got his data, he said, "Sure, keep them. Do what you want with them. They're like infinitely reusable." And we blew all three, and they're like $750 for three. So we're certainly looking to get some funding, and so we can continue this work. As I've tried some more classic organic chemistry techniques to see what kind of volatiles are in the medium, and they are just not able to concentrate it near to the level that's needed for confident GCMS analysis. So that's ultimately what we want to do. We want to get some more SBE fibers, and then we want to see what kind of molecules are arising in these really different media conditions. And then, to explore the gluconate aspect, we can use enzymatic tests. We can look for those enzymes that I showed you are involved in the conversion of gluconate, or excuse me, 6-phosphoglucose to gluconate. We could see those maybe pushed up in their concentration. And you can just analyze, when the bacteria is grown with the fungus, how much gluconate production is it doing versus when it's grown alone. We could analyze how much gluconate's making it into the medium. For instance, there's a lot of places you can go with the enzymatic test, but it's basically just to detect how much gluconate's around. Really, I want to use real-time PCR, which allows us to look at, for instance, what kind of mRNA transcripts are around, which tells you what kind of proteins are being made. I'd like to use the real-time PCR to really get down into the weeds and see, are these transporters being upregulated in these organisms? Are these particular enzymes being upregulated in these organisms at the genetic level? So I'd like to kind of end the talk there. I'll take questions after I go through this acknowledgments page. I'd like to thank all you guys for having me here to talk. It's certainly a joy to dive into this with interested parties. Because Dr. Overton and I we're super jazzed about this. Like, we can't get enough of it. We want the work to go faster, but working with undergraduates with budget constraints, it can be very tough. So the student support that you gave really made a huge difference. I mean, it supported now these 2 summer students, and they learned a lot about these systems and really skills that are going to help carry them in their careers. I gotta give a shout out to my friend Jared. I wouldn't have gotten into GCMS if it wasn't for him. So, anything that helps me expand my sort of expertise is always cool. And then for that to blossom into something more I could do with the students here, as well as colleagues, was great. And then, who's keeping the lights on and keeping the space cool for me. I'm dreading going home. It's going to be so hot. My office is so perfectly temperature controlled. Thank you, Commonwealth University of PA, for paying me and keeping me cool. So with all that being said, I'll entertain other questions at this point.
Well, thanks for a really cool talk, Kyle. Appreciate that.
You bet.
Where does some of this possibly lead to treating diseased bats or having this combination be less virulent? So can you, locate one of these chemicals that might help with that?
Yeah, right. So I think the idea, right, is, for instance, if we really understand that it is this sort of pathway that's being involved primarily in how they're able to be so prolific in the bat wing. Well, you can imagine, there's probably small molecule analogs of gluconate that could get stuck in the gluconate transporter and keep it from being operational. So if you block gluconate transport, right, bacteria can't acidify the medium. So other bacteria might be able to outcompete it. Those bacteria maybe are making antifungals that do hurt PD. There's no guarantee that these antifungals we see are that detrimental to PD. I think that, both of them are making the bacteria is making some antifungals, antimicrobials. The fungus is making some antibacterial antifungals, and they're keeping each other kind of in check. But yeah, we could totally get some kind of molecular analog, block these particular points within these pathways, right? And then doing so, you can treat, the bats somehow I don't know if it's through cave spraying. I don't know exactly how this work out. I'm not the biologist, but there's going to be a way in which having knowledge of how exactly is this symbiosis being powered, you know, it's just like this, right? If you have a blueprint of some castle you want to lay siege to, it's going to be a lot easier to take down the walls than if you don't, right? So this is very much trying to figure out where the chink in the armor is, where the weak point in the wall is, and then we can rationally think about how to um, how to combat this. Does that sort of answer your question?
Sure, thank you. Yeah, the little brown bats in northern New Jersey, where I live, were just totally decimated.
Yeah, and it's the big brown bats that have the ability to sort of resist the detrimental effects of white nose. And that's interestingly enough, when my wife was doing her sonar work, that's what we found. So the sonar could detect the difference between big and little brown bats, and this was a many year project, and you could track over the years, big brown bat signal goes up, little brown bat signal goes down.
Do the big brown bats migrate? I
No.
think that no.
No, there are three migratory species. So the red bat, now I've forgotten the other two, but they migrate down south. So they're not the cave dwelling bats like the little brown, big brown, and a couple of others. The Indiana bat, which is a federally endangered species. I volunteer at the Great Swamp National Wildlife Refuge, so I used to help out with some bat studies. Sorry, Dave, what was your question?
You answered it. The big brown bats do not migrate. I thought that might be why the big brown bats were supplanting the little brown bats, because they migrate and therefore not susceptible to the detrimental effects of the white nose. Because the little brown bats, I know, the ones that are affected negatively, they wake up out of hibernation and then they look for something to eat, and and it gets cold and there's nothing to eat.
So I think you're onto it there. I think I'm kind of remembering this conversation with Barry, in that big brown bats are bigger. They got more
Yeah.
fat, right? They don't need to get food as soon. So just body mass, they're able to survive that food drought, a little bit longer.
But the big brown bats are also affected by the white nose. They get
Sure.
the fungal infection. Okay,
Mhm.
thank you.
Yep.
Yeah, but not so deadly.
Hmm.
Right.
So is this a hygiene issue? Maybe not even just hygiene of the bat, but hygiene of their space where they're living?
Yeah, that's a great comment, because one interesting thing that Overton found was that you can spray the caves with polyethylene glycol, and polyethylene glycol will sort of put an osmotic pressure on fungus. It will cause them to, I believe, dehydrate. Um, I mean, that's what my sort of first principles chemistry thought of it would be. And by spraying PEG in these caves, he's found that the PD counts go way down. So I don't necessarily think it's hygiene per se, but it is a
Could
way
be
in
as
which
simple
he
as
can.
airflow then.
What's that?
Could be as simple as airflow then.
Possibly, yeah, possibly. It's
Awesome.
yeah, it's really interesting, but I think ultimately it's that they cluster, right? And that's going to, they're social animals. That's going to affect how this stuff spreads more than anything.
So the experiment sort of failed because of the melting filters, and they melted at only 37 degrees
Oh, no,
centigrade.
no. The GC port is 250 degrees Celsius, which these fibers are supposed to go up to 300, and they say in the instructions put them at 250. I think what it was more of a user error. So if you don't set the depth gauge on the cartridge itself correctly, you can end up touching the tip to a really hot metal part. So I think that, you know, Dr. Troy Dermata and I, who was one of the authors on the first screen, we had optimized, you know, what the safe length was. But I think that, you know, when the student was doing the injection, she may have not, you know, tightened the thing down far enough so that the tip would not be in contact with the sort of metal, um, entryway. Um, you know, we're trying to get it down into just a heated open space when we depress the syringe and allow the tip to come out into contact with the air. So it was a real shame. And I remember when the first one, you know, it took me, these fibers are really, really small, like they're hard to almost see. And it took me like we actually ran an experiment without a fiber. I'm like, there's nothing in this. And I pull it out and I'm like, oh, the fiber's gone. And then the second one, you know, she had put it in and I go in there and I'm like ready to pull it out because you don't leave it in there very long. And I pull it out and it's just goop. I'm like, oh, jeepers. And then the third one, you know, that one just again, same sort of story. So it was really unfortunate. It was really, really unfortunate because those things, like I said, they're like infinitely reusable, which is why they're so expensive. But yeah, I mean that's you know, science can fail because of bad hypotheses, right? But science also fails a lot just because this stuff is sophisticated. You know, it's not a, you know, it's like a professional sport. You got to like practice at it. You got to burn a few tips along the way. I don't know. But certainly, you know, having gone through similar things, I'm very forgiving when the students do destroy things. It's part of the learning process.
Well, isn't there some way you can create a guide to prevent that from happening? It shouldn't be a human, you know, dexterity issue.
Yeah, I mean, we did have you know, verbal, like this is how you do it, but ultimately, you know, students in a rush, they forget what you said. Yeah, I probably need to put down a you know, printout for the next round.
In other words, you can't build a guide to prevent it from going down too far.
I think it's more it doesn't go down far enough.
Far enough. Okay.
So yeah, it's just a matter of really making sure before the student does the injection that the distance is right.
Okay. Any other questions, anybody for Professor Root?
That was great. I learned a lot. Thank you, Professor Root, for that. That was really interesting, the technology, and I hope that the ongoing work bears more fruit as you go forward. And who knows where this is going to lead, but it was very interesting. Thank you.
Yeah, I think it's really exciting stuff. It has potential to be very splashy um and have an impact on people, animals. What could be better? So we're going to continue it. It's and and we have some ideas to get those tips again. So you know, that's what's holding us back right now. So I
Thank
appreciate the
you.
interest. Thank you guys.
So you haven't gotten involved in mushrooms after this, right? You're not going to make mushroom beer.
No, that's up to my buddy. I mean, I'll test it out for him. But yeah, I'm more of a fly by night mycologist in this case. I mean, they're very awesome organisms, of course, and you know, I'm uh, I don't identify them, but I love coming across mushrooms. I think they're beautiful. I'll take pictures. You know, I take nature photos, so I'll take pictures of them. I had this one photo I love. It's a, you know, an eastern box turtle eating an amanita. I'm just like, buddy, you got some good enzymes in there to be able to process that what is it, muscarone or whatever is so toxic in them.
Okay, well, thank you very much.
Muscaria, right?
Muscari. Yeah, I can't remember
Yeah.
exactly what it is, but yeah, I know that I can't eat it. It's not going to make me double in size like Super Mario or anything like that. So I'll let the turtles take care of it.
Yeah, it was a bonus, the experimentation with the beer. So that was interesting too. So,
I thought you
Yes.
guys would like that. Yeah,
yeah.
in lieu of a lot of data, I felt like, okay, this is very much part of the story and very much interesting for a mycology club to know that mushroom enthusiasts are trying to make beer out of mushrooms, right?
Yeah.
So it's
And
going beyond the
just to
kombucha.
kind of piggyback off that, it seemed like if I'm understanding this right, a lot of this presentation was above my head. It was really cool. I'm going to watch it back and learn some stuff. But it seemed like the takeaway from the beers that were brewed with some percentage of fungi presented some interesting smells that weren't present in the control. Is that what we can take away from that?
Yeah, absolutely. Yep. Yep. Not all of them good.
Yeah.
But I do think it's promising. I think the lower percentage, like 10%, those ones seem to be very similar to the control. So I would tip one back without worrying too much about the baby diaper.
Okay, well, you're welcome to stick around on the Zoom call if you want to watch the mushrooms being presented.
I actually may hit the road. I haven't eaten dinner yet, and gotta let my dog out
Okay.
and go roast in my house. But hopefully I'll see you guys again for the next chapter to this work because it is still ongoing, and I have a lot of confidence that Raymond and I will be able to show some interesting stuff going on with the symbiosis and the presence of glucose. Yeah.
Okay, thank you.
Well,
That was great.
give our thanks to Meg and all of your collaborators. It sounds like there's a lot of learning being accomplished there, and that's the objective. So,
Absolutely. Yep. So I appreciate you guys too. Thank you so much. Take
Have
it
a good
easy.
night. Okay, as we turn to the second half of the hour, or 2 hours, if anybody has any mushrooms to show, if you want to put your name into the chat.
Igor, I was hoping I'd hear some questions out of you for the chemistry there.
It's just routine sugar biochemistry. I was never particularly fond of sugars. I saw the beta glycosidic linkages, all these ketones and esters and all these alcohols, and I'm like, okay, all these chiral centers. If you saw the lines that were dashed, that were solid, those were chiral centers. Meaning that there's a lack of symmetry in there, and because of that, you have two sets of molecules that could be mirror images of each other, like your hands. They are not superimposable. You have the same structure, but they are mirror images of each other. So yeah, that was fun, but it was fairly basic.
Yeah, that's what I was thinking. Not.
Yeah, I mean, the kind of reactions that those enzymes perform, it would take like chemists a few steps. There's one reaction that you cleave a carbon unit from going from a 6-carbon molecule to a 5-carbon molecule, and then you oxidize the middle alcohol to a ketone and leave the outer one as an alcohol. And like you have to do a number of steps to accomplish that kind of thing. If even that. So that was fun. It's like a magic stick. You touch this, you go from A to B with a magic stick. And your magic stick is being the enzyme that is like this huge protein that's coded for by multiple genes or whatever. So yeah. So no questions. I mean, I got the idea. How see what the balance is between bacteria and fungi, how they control each other, and through that gluconate molecule. So
Dave, in the chat, I put the 3 migratory bats.
Oh, okay. Yeah, you mentioned a red bat, an Indiana bat, and
No, the Indiana is non-migratory.
oh,
I-
okay, I should look then.
I mentioned it that it's federally endangered.
Oh, I see. Are the little brown bats now endangered?
Close, yeah, well, their population dropped to like 95%.
Right. Yes,
So
I
they
know.
certainly threatened, that's for sure.
I wonder if the ones that survive might constitute a more genetically resilient um, uh, breed of little brown bats.
That's what they were hoping. I haven't kept up with it, but I did a trifold, I don't know, about 5 or 6 years ago.
In the caving
So-
community, they keep people out of the bats. They always did to a certain extent because of the hibernation, right? Don't disturb them in the winter, but once the destructans was detected, actually in Schoharie County, New York, they really got into higher levels of protocol in terms of sanitizing the cave gear and access and whatnot. And I think some caves have never been reopened actually to protect the bats, but it's probably going to spread everywhere before the tide turns.
Okay, I'm sharing my screen because I do have a first mushroom of the year for myself. I wanted to ask if anybody's had problems of uploading to Mushroom Observer, because I had lots of problems in the last 2 days just trying to get up.
Were you trying to upload from a phone or a computer, Sue?
From a phone where my pictures are.
Okay, so could be some problem with the software. I'm going to ask Joseph
I
Cohen.
already
Oh, you already
exchanged
inquired.
emails with
Sue, I have a
support.
suggestion for you.
Yes.
Put your observation on INAT and then use the import from INAT feature on MO. It's really fast. And then you
Yeah,
get
but
2
it's like
observations for the price
if I
of
like the
one.
interface for INAT, I would be on INAT,
Oh, I see. Okay.
Oh,
right?
I was going to say, I prefer but I was going to support Dave on that because it does work like a charm.
Yeah, I did try the application for MO, and well, it did get the picture up, but the rest of the interface I don't like because it doesn't have my templates that I have on Mushroom Observer.
Oh, you were using the MO app.
I no, I just did that once because the browser upload was not working.
So, Sue, what did Joseph tell you about the problem?
No one else has reported any problems.
No one has reported any problems to them, but you.
Yeah, yep,
That's
yep.
bizarre.
And he says, well, he's going to dig out. I'm an Android person, so he's going to dig out an Android. But I don't know if it's the operating system. It could be because everything's been updated. The operating system and the Chrome constantly change. So anyway, this was my first observation as a drive-by on a car. Because I had to walk 20 minutes to go find it. I did not expect to see this as I was driving by. It's the jack-o'-lanterns, and I did not know that they would appear this early in the year. But apparently they can be seen late summer, even though I don't consider this late summer yet. Anybody else seeing jack-o'-lanterns this early?
Nope.
Early July. I've seen them in early July, but this
Well,
is
we're pretty
earlier
close to
than
that.
earlier. Yeah, this is earlier than usual, even for even for you, I guess. They're finding black trumpets here, though. They've been finding black trumpets for over a week near where I live. So there are some things that are coming out early, and black trumpets are mycorrhizal as well, mostly with hardwoods. These are saprobic.
Right.
or parasitic possibly, but I think saprobic.
Well, these
Probably
were found by some young spruce, but since it's a fairly new park, less than 10 years old, I'm sure it's other wood that's underneath that, not the spruce.
Yeah, they usually jack-o'-lanterns. Well, the typical species, the Omphallotus illudens, is generally grows on oak, but it grows on the roots often, and often from buried roots. And you don't even see a tree, and you'll
Yep.
see
Yeah, we're
jack-o'-lanterns
mostly
in a
an
yard.
oak area up here, but like I said, this
Mhm.
park is new. So I think they just removed the old oaks and put some young spruce there. Does anybody else have anything you want to show? Or should we start with Hunter? We're-
I've got one little one.
Brandon says he's got some stuff in the chat.
Just wanted to chat. All right, let me stop the share. You want to go next, Chris? I didn't see this.
Yeah.
Anybody in the chat.
All right. I just had this one. It was in my garden, right on the edge of my wood chip walkways and the actual soil in the beds. And it was just at first I just saw all of the stipe like peeling away like that. And then when I dug it up, I think the most interesting thing about it was this mat of mycelium. This is all like, at first I thought there was maybe like a stick in there, but I like broke it in half, and it's just a solid mat of mycelium, like hard to the touch. But just some fairly regular spores. Not much of a spore print came out of this thing. It was a little on the dry side, but some cool Basidia everywhere. These little flat topped cystidia that couple came out.
Did you
But
propose a genus yet?
no,
Well, did you mount that stuff in water?
yeah,
Or
the first two were in water, and then
looks
the
like the
other.
spores are pigmented.
Yeah, what color was the spore print?
It didn't even lay down a spore print. It was I don't know, it just wasn't pushing them out. I still have it sitting on the plate. I'm pretty sure there's still not one.
Do you have an idea of the spore measurements?
I didn't measure them, but all of this structures are pretty small. This is pretty zoomed in,
Yeah,
even at 400.
yeah.
Can we see the gills?
Oh, did I get that picture in there? They were like a smoky, pretty tight, packed in there. So I was thinking it would have probably had some kind of darker spore print, but it was hard to tell. And even that, the edges of the gills were starting to dry up a bit. It was
So
just,
that just from the way the mushroom looks, I suppose, and the pigmented spores, you might suppose it could be like an agrassibe because a lot of them, several different species come out in late spring. But those small sort of truncate cystidia, those are kind of unusual. I don't recognize those as agrassibe cystidia.
yeah,
And the small spores.
I'll have to
There's a
take some measurements.
Yeah, most Agroscibi have spores that are at least up to the maximum length is at least 9 microns. But I think there's 1 or 2 species that have small spores.
Yeah,
But yeah, that's just a really a guess based on not a whole lot.
yeah, I was just amazed by the how tough that chunk of mycelium was on there. Like it just seemed odd to pull out. And it was just like it was holding on to, not just a bunch of soil, but 9 different wood chips. It was like it didn't want to let go of any of it. Robust. That's all I had going on there.
And that would not be a sclerotia. A sclerotia would be much deeper, or would that be one? Does anyone know?
Hunter, where do you think you see a sclerotium?
I don't. I mean, he just said it's like this very hard mass of mycelium.
Now, sclerotia would be small and,
Oh,
more or less roundish things.
okay. Deeper down too, or not necessarily?
Yeah, there's one species of Agroscibi or Agroscibi that grows from sclerotia, and I think they do tend to be deeper than an inch, or maybe as much as 3 inches below the surface. I forget the species name, but there is one Agroscibi that does fruit from sclerotia, and it looks a little bit like this thing too, as if I remember correctly. Let me look. I'll put a name in the chat.
Are there mushrooms that have rhizomes? Or no?
Yes, most Agroscibi has rhizomes, as do many of the other Strophareaceae, I believe is the family.
Armillaria, right?
What's that?
Armillaria.
Yeah, Armillaria's in it. I think they're in a different family, but they have those really brutal rhizomes that grow underneath the bark of a living tree and kill it.
The rhizome would be bigger. I mean, this one looks more like a rhizome to me if I compare it to flowers.
Compared to what? I'm sorry, what did you say?
I compare it to the mass of mycelium looks like a rhizome to me. Because flowers have rhizomes, I think, reproduce that way. But I haven't looked at rhizomes in the mushroom world.
I think it's just a really dense mycelial collection, right? If you are cultivating, you've got, oysters grown in a bag or whatever, it can get really, really thick and dense like that in the wood chips or the substrate. But
Yeah, there's no shortage of food for that thing they grow on. That's for certain.
yeah.
All right, Brandon, you want to show something? Or more than one?
Yeah, sure. And I'm unfamiliar with the rhizomes in plants. I'm coming from natural sciences kind of backwards, learning about fungi first and going from there. But I would agree that was a bunch of tightly compacted rhizomes, not necessarily a mycelial mat. You know what I mean? But let's talk about what I'm calling Xanthoconium affine. I really want to talk about the variations because I think that's what this is. So can anyone agree or disagree whether it's Xanthoconium affine?
Where was this growing?
Say it again. Where?
Where was this growing? Yeah.
So this was in Pine Barrensy.
Pine Barrens.
Yeah. With mostly oak, hardwoods at least, oaks. But there were not a lot of conifers in the area.
Yeah, this type is very typical of Affini or Affine, how you want to pronounce this.
Affini, yeah, Affini. Yeah.
Aphany.
But it's not reticulate, right?
No, it's not supposed to be. It should be
So,
glabrous, kind of a little bit translucent, like an ivory. Uh,
and I wouldn't call this the spotted one. So I
no.
would call this affine variation, affinity variation affinity.
Yeah, if there's such a difference. So in the image, the cap is kind of a slightly purplish, maybe. So that's probably just an illusion, right? The cap was
Yeah,
brown,
yeah, it was a trick of the light. It was
right?
very dark, but it was brown. It was more this color, and then I don't know. My phone's been doing some weird AI stuff recently. I didn't pick up on any purple in real life.
Right, but yeah, I think it is Aphany. I don't think this is a Telopolis. So
And all this damage here, it looks physical, like it looks like physical damage. To me,
right.
I don't think this would be one of the affinities. Uh, so
No, this is clearly like
maculosis.
not like intact uh, pellicles. Something either was rubbing against it or something got eaten away. Yeah,
It's a very sluggy year.
doesn't look like slug damage, but yeah.
I just got back. I put something in the chat. Agricidae vivaceae, I think. Anyway, it's got small, rather somewhat small spores. It says they go up to 9 microns, but without a distinct germ pore. And Christopher's spore shot looked like spores that did not have a distinct germ pore, which most Agricidae do. So anyway, you can look that over. I'd say that this is Xanthoconium aphany. Looks like it to me. It's just a dark one. Sometimes they're dark cap. The cap color is highly variable on this species. I see a whole lot of it. Sometimes it's among the first species of boletes to appear around here. No staining, right? No staining on any parts, except if you scratch the pore surface, it might just get a little light brown, but that's just
Yeah.
because of you're compressing the material.
Yeah, and Dave, do you have any opinion on variety here? I was calling this variety affini, Xanthoconium affini, variety affini.
Well, I think that's the one that's got spots on the cap. Does it? Are those?
No, that's
There's a couple
No,
of
that's
little white.
maculosis. Yeah.
Yeah, that's maculosis.
Oh, that's maculosis. Oh, I'm sorry. So this would. Oh, I'm sorry. Duh. Okay, so this is Aphini is one with no spots.
Yeah,
That's what I would say.
I would say this is Aphini. Yeah.
There's another one too that's sometimes reticulate. Is that Igor? Do you know if that one is a distinct variety or is it just like an
I don't know.
anomaly?
Like I said, I suspect there'll probably be no difference, genetic difference to even, start proposing whatever, subspecies or, yeah, varieties. For what it's worth, I mean,
Okay.
if you like them, keep them. So I'm not
No,
gonna
so
object.
so you're
Yeah.
saying like even the spotted cap variety, the reticulate variety, or affine, if you sequence them, they'll all just kind of come back really, really close.
I would say so, but again, we have some interesting, challenging questions about some fungi, like for instance, Hortiboletus flavorabellus, where
Right.
yeah.
I
So I mean, I don't want to say anything before actually I see evidence, but yeah, the subreticulatum is a variety, and I don't know if that comes together with the spots or without the spots. So yeah, you have to look for that kind of thing in the field. So it could be a real thing.
Yeah.
So that's
I
the
am
category I study these
guessing.
varieties.
I am guessing that these things are all going to have essentially the same DNA. That's why they're varieties, right?
Yeah.
Yeah, so variety is not really like a subspecies where you could actually
Right.
see some kind of a phylogenetic structure to a clade, and you can say, okay, we have different groups here, different populations or whatever. So you can start proposing maybe subspecies or whatnot, depending on what your biogeography is like. But yeah, for variety is a less stringent, taxonomic unit, I think. So if it makes you happy that you have a spotted variety and a subreticular variety, that's fine. Whatever.
If they're not buggy, go for it, right?
I propose maculatus all the time when I see those that. So
All right, and we got a late start. I don't want to take up too much time, so this is the last one I'll share. I shared it last week, so Scutelinea, but I put a name to it. Vitriola, Vitriola, and I'm calling these broadly ellipsoid, which is a distinction with the spores, and it is kind of tough because I don't know, broadly ellipsoid, narrowly ellipsoid nonetheless. This is at 400. The spores were at 400. This ascus is at 400 as well. It's not a small thing. Approaching 190. This one, I saw a couple of them that probably exceeded 215. I put the average. Actually, I'll go through all those numbers when I get out of these photos. These are the marginal hairs, which is something you have to look at for these things. Huge. This is at 100x. So, call that 800 microns pretty much. This one that you can follow the whole way up. This one is a different one that is larger than that. I measured a bunch of them. They were between 650, but I kind of want to discount some of those 650 ones because there were these also intermediate hairs. But the big ones were all plus 800. So that was kind of on the smaller side for the marginal hairs. The biggest one I found was 950, so less than 1,000 microns. These are the hairs from the underside, much smaller. This is also at 100. So, 1, 2, 3. This one here that's kind of in focus, somewhere around 31, 32, 33. And then something to note that you could see on the previous photo on this hair here is that this one has 2 feet. It's kind of, some of them have 2 or 3 feet. Again, just to show how big these hairs are. This is at 100. And if you focus on this one here, we go zoom in a little bit, you can see it's at over 90. So 90 integers, that is. So this one here is 920 probably microns. And also note that they're septate. You can see all the septations here, and they're not just 4 septate. They're each one is at least 6 of the big ones, which is another thing you want to look at. Also, look at that. That one's got 3 little feet. The faint one in the back there, if you can make it out. This is showing those feet again. This one here has 3, it looks like. This one obviously has 2. Maybe a third one's coming off of something we can't see, but the distinction there is, does it have 1 attachment point, or does it have 2 or 3 attachment points? And that distinction really actually is it 1 or is it 2/3? Not is it 1 or is it 2 or is it 3? That second option is 2 or 3.
So there's some of them, and I just had to leave the room for a minute. I just got back, so we're looking at Scutelinea, right?
Yeah.
And so there are. I haven't reviewed this lately, but there are some species for which the hairs are maybe bifurcated or trifurcated at where they attach at the bases.
Yes. And
okay.
it's an and or situation. So
yeah.
You
Yeah,
know, if you're keying it out.
The
If you're keying it
inclusive
out.
or. Yes.
Yeah.
Did you know what
Cool.
for these things, a key, here we go. Was this from a spore drop? Yeah, because a key feature when trying to identify Scutellaria, which is very frustrating, is mature spores, the ornamentation of mature spores. How much is there ornamentation? How much of it, how coarse is it? Is it a reticulum? This one looks like it's kind of grainy looking, warty.
Yeah, it's not a good photo. This is at 400x. I'm taking it through my phone, which when I get one of the ridges in focus, by the time I get my phone focused on the spore, it's already out of focus, if that makes any sense. But this, I could see it with my eyes. And thanks for talking about the spore and ornamentation. I think I forgot to talk about it. One of those keying features is, what is the spore ornamentation? Is it warty with big warts? Are those warts reticulate? So are they connected? And I called this medium warts, some reticulate. And that's what these ridges kind of show. These dark ridges kind of show how those warts are connected a little bit.
You might need to mount in a stain to
I
see
should have mounted.
if there's a pattern to the ornamentation. I'm not sure about that. You might want to do some reading about that. But the ones that are reticulate, there are these good sized pits on the spore that are just arranged, that are surrounded by ridges, and the boundaries of the pits are the ridges, and those boundaries make a reticulum, and it can be pretty obvious on some types. So, is this some different species here? This is the species.
This is the species that I applied.
Oh, okay. Yeah, because this looks like your spores. Were the spore measurements the same?
Yep.
Wow. Because that's also important too. There's a fair amount of variation on spore dimensions in genus Scutellaria.
But you can see here how that looks a lot like and obviously I couldn't make out the best detail, but it's, the warts are present, they're medium, and some of them are reticulate, but not all of them, right?
Well, there's some ridges.
Some ridges.
There's some ridges connecting.
Yeah. Correct.
And look at they're showing bifurcated hairs, the bases, and are they showing any they're showing one that's, some of them are trifurcated.
Mm-hmm.
Some of them are kind of bifurcated and then bifurcated again.
Mm-hmm.
But that might be really hard to see.
Well, I have a photo of it.
Oh, did you find some hairs that were bifurcated and then the bifurcations were bifurcated? Because
yes.
Boy, that would seem like a pretty strong feature.
I think I showed it. Where did I show it? Yeah, this one here.
On the right there.
Yeah.
Oh, I see. Oh, I think I see that. Right, the one, the split on the right part, pointing upward, has another split. It looks like.
Yep.
Wow. Yeah. So if you think you know this species, you might be able to post it online and not have Malcolm tell you why you're not able to really know that. That would be quite an accomplishment.
Well, yeah, this is the name I proposed, Vitriola.
Vitriola. Wow.
The spores don't look right on Patagonica.
Looks like the Q is wrong for these guys.
Way more broad, right?
Yeah. Yeah.
This has potential. I forget why I ruled it out. The edges, maybe, well, spores fit.
That last spore you showed.
The hairs, the hair length is really how you tell these things. I think in my observation, see here, 1,200 to 2,300, huge, massive.
That one spore you showed
none-
also looked like it wasn't all that much different from smooth.
Yeah. All right. Well, just wanted to show that. I don't want to take up too much time. We only have 18 minutes
No,
left.
that one was worth spending some time on. 'Cause there
Yeah,
was
Nice
a lot of
work.
I'm
little
glad you
details.
found this other site. My fungi in the UK, in the Yorkshire, is very
So
small
this is
site. You wouldn't have thought you could find something useful there.
so the person that we keep talking about, Malcolm Greaves. It's this guy here. It's his key, and he's pretty present on INAT. It's an interactive key where you can just look, plug in the spore length. I had 19.9. Sure, my spore widths I think were somewhere between. I can go back and look at it all.
The Q looked like it was
Yeah.
at least close to 2, maybe a
Yeah,
tad
it's
under.
18 to 22 by almost exactly 10. So that's 2 in my
Yeah, right
opinion.
around 2. Mhm.
But you can plug
Okay,
all
but
these
matching
values.
it would be fair to match the European species. Do we know if it's supposed to be here or?
So the closest thing, yeah, if we want to really get into it, the
I know we
things
could
that are
probably spend the
being
whole half hour.
well, yeah, the things that are
Yeah.
being proposed as vitriol here in the U.S. and it's not very common they've been sequenced and it's an Indiana number, right? It's Scutelinea Indiana 05, I think it is.
Okay.
But you know, even Malcolm, who's in the UK, that one's never been found in the UK, but he still knows the thing. Yeah,
Okay.
not yet found in Britain, France. So he knows his American species too.
Perfect.
Yeah. Thanks, guys.
Yeah, I lost my Wi-Fi for a second a little while ago, so I don't have the chat. But anybody else in the chat, or should we move on to Hunter, who sent in an email?
Dave, did you want to go first?
It doesn't matter one way or the other to me. You go ahead if you want.
Okay, I'll go quick. I didn't do any microscopy or anything, so I'll just make it quick. I did want to say the first trumpets I ever found was on June 4th, here in Illinois, and then I just found some this weekend as well. So I think they have a long season. I think it's just condition based. But anyways, this I found, I think it's Amanita multisquamosa. I've got a couple more pictures. I have this specimen. I'm trying to get some spores out of it right now. It's got that rolled sock appearance on the bulb and striation, pretty striate on the cap. And that's about all I got to say about it, really.
Unfortunately, we can't check Ratulas's website. It's, as far as I know, it's down, but I believe that he described multisquamosa as having long striations, which this one appears to have.
Yeah, I read up like 50%. So it's close.
Those are some really big warts on the cap.
Yeah. It's funny, the phone camera really sometimes I noticed on my chrysoglemas lately that I was like, wow, those are really popping, and it didn't seem as much so in real life. So I don't know, but yeah, pretty pronounced. I mean, so this would be in, would this be like the Pantherina's, would it be?
Yeah,
What-
is it series Pantherina?
I think that was the last way that classification was proposed as a series, which I don't even,
subsection.
I'm not even sure what that really means.
Yeah,
But yeah,
me
I think so.
either. But yeah,
This would be in that group?
Yeah, mostly based on the rolled sock basal vulva.
Okay, cool. So it sounds like people agree on this, probably what I have. We can move on.
well, I was saying series Pantherina. I
Oh,
often
there's
do.
another thing to check also.
I meant that as a question.
Um,
Some of the series Pantherinae also have the inverted funnel annulus. That one had an annulus, but I forgot to look. But this next thing though. Oh, I see. Okay, no, I was going to
And
say something, but no, I'm not
on,
going to.
on that note with the annulus, because we talked about this kind of last week with the thing that I found, I think it was after maybe, what was that other thing? Subphalatip.
Velatipes and
Velatipes.
subvelatipes.
Yeah.
They are very similar.
Another thought, yeah.
This one's colored like multisquamosa, but I'm not sure how important that is because when you start getting these things sequenced, you get some surprises. And this annulus is not really pointing up, but it looks like it may have been before it margin kind of collapsed a little bit.
Yeah, it does.
Sorry, we should move on to the next one, really. Yeah, I just had that quick question about the annulus.
Okay, I do have the specimen. I mean, is that something we're sending somewhere or no?
Well, I suppose it depends on whether or not you have are involved with any sort of project or anything where you can submit a lot of sequences and get a decent price per sequence. So
Okay,
it's a judgment, value judgment.
okay. So I think this is ready, but we just crazy is I didn't even do uh, um, a longitudinal cut. Um, it's pretty young, and I do have the specimen, and I'm thinking it's this mostly because I found something very similar in the same spot before that I labeled this. And, I just didn't have time to go too deeply into IDing it. So I don't know what other people think.
Is that a wine brown stain on the base?
It looks like it. I'm gonna grab the mushroom right now.
Igor, you're still here, right?
Oh yeah.
What's that one with the wine? Is that Vanessa seps or something like that?
Vanessa peas. Yeah, this
Vanessa
is
peas. I'm sorry,
this is that.
not
This
the
is
head.
not that.
Vanessa peas, right?
Yeah.
But
Vanessa.
this looks too pale for that.
No, this is Regius. No question about it.
Yeah.
Oh,
Okay.
yeah, and it has a real foot. When I look at the specimen now, it's kind of coming straight forward, so you can't really see that it has like a hook at the bottom.
And hardwoods, right?
Yeah. Yes. Yeah, oak, hickory, you can see the hickory nuts. Yeah, no conifers. Lakeside. Small lake.
Okay.
Okay, cool. So good. And then I know last week Dave had some small marasmias, and we were talking about some of these little tiny white guys. I think this is Tetrapurgos nigripes.
I think so. Did you get any spores?
I tried and I didn't see any. What color are they supposed to be?
They look like jacks. You know, like jacks that you,
Oh.
when you're a kid and you bounce a ball or whatever and grab the jacks or whatever that.
Yes. Okay, you know what? I'm gonna do
They
like
look like jacks. If you smash them on a little bit of gill, you're probably going to find some of them.
yeah, that's what I'll have to do because laying one of the caps down, I couldn't see any spores
Right.
at all.
Yeah, that happens.
Okay.
So I forget about whether or not that this species has these widely spaced gills. I just forget, you know. But the other photo certainly looks like this Tetrapagos. I forget the name of the
Yeah,
species.
or there's the alba is the other one.
Oh, okay. Oh, so they might both have those spores shaped like jacks. So
Yeah,
it might
either way, if you do a smash mount and find some of those cool spores, please share them next week. Because
I will do it. It'll be fun to try to make that happen. Yes, I will work on it. And I mean, these are very small compared to, I have some pictures of this, what I thought was this species and labeled the species that were much more substantial. So I was like, oh, well, maybe it's alba. But then I read that alba comes out of like a little mycelial mass, kind of like what Chris was showing. And these come out like basically straight from the piece of wood they're growing on. And that's how they look to me. I can look a little more carefully with a little more magnification, but that was one of the main differences that I read. It's funny, like, Dave, you mentioned something about a collar last week.
you don't see that at all here. That's a marasmius thing.
It's funny.
These gills are subdecurrent.
Okay.
Adnate to subdecurrent.
Yeah.
The collar on it, like Marasmius rotula. Just look online for some pictures of Marasmius rotula. Or go to Mushroom Observer or INAT and look through observations. I know on my observations of Marasmius rotula, I will often include a photo showing the collar. So what the collar is, it's as if there's this tiny tire surrounding the top of the stipe, and the gills attached to the outer part of the tire. I mean, that's sort of what it looks like.
Okay, got it. Okay. And are they free gills then? Those morass?
They describe them as attached to a collar.
Oh, okay. Okay. Thank you.
Usually. I mean, you could call them free because they don't meet the stipe, but it's sort of a special sort of gill attachment.
Okay. And then I think I have maybe one more, and then there's a couple of pictures in the actual email. Nothing big though, so we'll fly through. Oh yeah. Yeah, one more. Oh, okay. So this is, I think, Lactarius folimus, and it exuded a ton of milk, stained brown. It didn't really smell fishy to me, which I think it's supposed to. I thought it smelled like cut fresh potatoes or baked potato even on my hand. And I still have them. And that's what they are though, right?
Did you nibble one and see if it was mild tasting?
No, I didn't, but I still have them.
Yeah, I mean, look at all that milk. Oh, and I see some brown staining on the
Oh, they really,
underside there where the milk's coming out near this. It looks like it's staining the gill brown a little bit.
Yeah, it's stained. I got it on my pants, unfortunately, and they were like light colored, and now they're yeah stained.
Yeah, in areas where there's a lot of these and they're popular edible, people often pick them wearing rubber gloves.
Mm-hmm.
What I've read, I think I read that in Bill Rudy's book.
Is
I'd
this
love to
Salinas?
try to find a lot of them. I never really do though, not a lot, But
But
these
that's
really,
interesting. There's no odor because these really smell like fish. You
yeah,
know,
not fishy, like baked potatoes. I thought, but my smell could be off right now. Yeah, but
you
they
know
were
what?
so,
If you get the Bisset Lactarius book,
yeah.
Bisset Harris Lactarius book,
I don't have.
there's a few varieties of Volimus, and so there might be some variability
Okay,
in
I'll
the
let you
odor. I'm not sure.
What bothers me here is that the gills are white, as if it's almost like afflicted with some kind of a mold.
Yeah,
know.
those are really pale gills. You're right. Yeah, usually the gills on
Green.
Volimus
And
are a
I
little bit
think you can see the
yellowish.
mold to the left. The gills are sort of getting smudged. It is like they're being taken over by something.
Really?
Yeah.
On the
So
stipe, on the top of the stipe there, it looks hypomyces or
it
something.
looks fishy. It looks milky, messy too. Yeah, I
Very.
think this one is definitely afflicted by some kind of a mold.
Oh, okay.
Yeah.
Well, I
So
won't eat.
I hope you didn't eat that one.
No, I didn't eat them at all, and I didn't even taste it. So,
That might be why it doesn't smell the way it's supposed to.
yeah, maybe. Huh.
Yeah.
Okay. I
The-
thought they were super fresh, but and I don't know. There was a lot of hypomyces out there on things.
So the gills were white like this. It's just not the camera just
Um,
didn't
you know,
wash
I
out the
thought they were
color.
a little more creamy yellow. Personally, yeah, I was just trying to see. I think I had another picture, and I have them in the refrigerator. It didn't seem like they were moldy, honestly. It seemed like they were super fresh. Um,
Well,
but
they look kind of yellow here. The gills on the farther away through fruiting body. So that color is probably correct. But that particular fruiting body in that picture, it looked strange to me. Not just the color, but there was like a loss of structure on the left hand side and farther away. It's like almost the gills were being taken over by hypomyces.
Yeah,
yeah,
yeah. Lactarius Volimus has really nice, even
You see
edged
them?
gills.
You see that? You see, like, it's almost
Yeah,
like a blanket there. There's like no gill structure.
yeah.
it
It's
couldn't just be like milk poured in there.
nah, I don't think so. Not that much milk.
No, if this was a whole lot of milk on the gills, it would be really brown
That's
in
interesting.
Volimus.
Yeah, yeah, okay. Okay. Well, anyways, they were beautiful. I mean, the orange on the caps was really stunning, actually. It's real nice to see. So anyways, I'm I really would like to try to eat some more lactarius. I haven't, I've eaten so little. I see what you're saying though. It's like the gills aren't even there anymore in some places. Yeah. Okay. Well, that's that. Um, and I think that's it. We did all three of the INADs.
I
Did we?
think we did all three, right?
We did the Amanita,
The Retabolitas and the Tetra.
we did it.
Hydra.
Okay. Thanks, Sue.
Thank you. Dave, it's about 9 o'clock. Do you want to do any, or you want to save them for next week?
I can do them. I'll have more for next week. There's so many mushrooms right now. It's ridiculous. I mean, I have so many photos from last week. This one is kind of um, I'm pretty sure this identification. I didn't do any microscopy or anything because there was only one fruit body coming out of moss that's covering a small patch of ground on my property. But I'm pretty sure the identification, Rickenella swartzii, it's not a very common mushroom. It's very small, like Rickenella fibula, and it has the decurrent gills like Rickenella fibula. Um, and it's got the stalk that's a little bit dark toward the apex. I mean, it's really it's like the poster boy for Rickenella schwarzii. So I'd be completely surprised if that's not what it is. But I put it right into the dryer. I wanted to preserve it and get it in the queue for sequencing later on. That was a pretty good find, I thought, because I've never found this species before.
I found things that I have called Schwarzii that I'm not very confident about, but that I agree that looks pretty textbook, man.
It really matches a lot of the photos and descriptions that I've run, that I've seen online. And it's very small, like Rickenella fibula, grows in moss, and I'd be astonished if that idea is incorrect. So this thing's pretty cool. We had a foray on last Saturday at the Pinchot State Forest in the Poconos near Thornhurst, and there were a couple of Boletus edulis. There were some really, really nice Neoboletus subvolutipes, but quite frankly, this thing here is more interesting. So I've never found this species before. I think this is correct. The spore size fits. There's another ribbed stocktail vela that has the color's a little bit different. It's more yellow than these gray ones. But the spores are a little I forget, bigger or smaller. It's one or the other. But the spore dimensions on my wasn't my collection. One of our members found it, and there was one really, really big spore in one of the pictures, and I thought, wow, that's kind of weird. It's not this one. One of the photos, I forget which one. One of the photos shows a spore that's like way bigger than the other spores. But I looked up the species, and in fact, the range of lengths of the spores is quite large. So that was not out of character for the species I proposed. Some of these spores look like they're almost globose. I think that's probably because they're standing up on end. I couldn't get a spore drop out of anything. I put the two best fruit bodies into the dryer. I wanted to preserve them. So I took the oldest piece that had some of the hymenium, this yellow part back here in the back. It was a little older. And I was not surprised I didn't get a spore drop. It looked like it was too old. But I got spores from a smash mount. And so when you get spores from a smash mount, they tend to not lie down very uniformly because there's a lot of material, causing how thick the layer of liquid is, the mounting liquid, and then the spores will have more of a tendency to stand up. But anyway, I thought that was a pretty cool find.
What did you call it?
Helvella costifera.
Oh, okay, it's not the cerebellum.
Yeah, well, there was one other species that sort of fit, but what's the name you're saying? Because that sounds like maybe one that I didn't check. So if you know another
That's
idea,
the one, Hunter. That's the one is that the one you shared? Was it last week?
Yes,
what's the name of that one?
I think it's a cerebellum.
Oh, it starts with A, right?
S.
Oh, it's S. Set SETA.
No, A. A is an apple.
SAT.
No, A is the first letter.
That's what I asked. Actually, yes, starts with A. Yeah, I think I looked at that one, but I'll check again. I think that's the one that's more yellow, and the spores are a little different size.
Okay.
So, one of our new members likes to look around for really little things, which is it's always nice to have a few people on a foray who are going to do that sort of thing. These were in moss, probably sphagnum, near a little stream, very wet area. You can see they're very small. So I think that they might be like some sort of hemimycena. I didn't do any analysis on them. because I wanted to just preserve all the material. I mean, all of the material. Okay, once it's all of these are dried out, you can practically fit all the caps on a match head. So I didn't want to use any of the material for anything else other than drying them out so that I can save them. So seemed pretty interesting, whatever it is. So
Did it have any smell?
I didn't check, but you know what? They're so small that, I was afraid if I tried to smell them, I'd just inhale one of them. These are tiny. I had to handle them with tweezers all the time. I put them into a bottle cap inside my dehydrator, which is a trick I learned from John Plischke for drying little things like this and little samples. They're pretty cool though, whatever they are.
Dave, what did you say that little trick was inside of your dehydrator? Did you say you put them?
I think a bottle cap, like from a beer bottle or soda bottle or something,
Oh,
and just put the clean out the bottle cap, make sure it's dry, of course, and then you put these tiny little things inside the bottle cap and put the bottle cap in the dehydrator
so that they don't fall between the.
Yeah, so they don't get blown around because the air current inside the dehydrator will move little things like this around. You're going to lose most of them. I think I lost one anyway. When I tried to pick it up with tweezers, the stipe just broke and the cap went flying and sort of like forget about it at that point. I mean, we're talking about, the size of a grain of sand at that point once they were dried. It was pretty cool find though. And here's another thing that I don't know what this is either.
Hey.
This is a white pine cone. So presumably white spored. I think I did take one of them and set it out for spores and I didn't get a spore drop. So then I had so much material I wanted to look at. I went out with some people on Friday and then we had a club foray on Saturday and I ended up with really so much stuff to try to look at. Looks like these gills are intervenose. So there's veins right on the gills and probably going deeply into where the gills are attached to the underside.
Wow.
But I don't know what this is either. So it's just another thing that I preserved and it's in a bag now. And so next time we have a rainy day, I'll be busy creating labels with INET and MO numbers on them for the. God, I've already got like probably well over 100 things preserved.
Wow.
Is it not Strobilurus?
Yeah, that's a good place to start.
Oh,
right?
Strobilurus.
Strobilurus?
I don't think so, but
That's
look.
Norway spruce, I think, the Strobilurus.
Yeah, there's one that grows on pine too. I should check that out.
Beospora.
Yeah, those aren't Beospora though. Beosporas are. These were kind of like resilient, the stipes and the caps with the material, the context is kind of resilient. Those Beosporas, they're kind of small but I'll check. That's an idea. I should at least check some of these pine cone type things.
Yeah, Strobilurus is, it's not just one species. There's a few species.
Yeah, there's a few species, right? I should go through that list. All right, I'm going to write it down. Yeah, I've just had so much stuff to look at that, one reason why I come here is, to get some ideas and then I saved myself some time. Oh, that gee, I think it was somebody here last week or 2 weeks ago. I had this like red slimy stuff on a log. And somebody told me what it was. I forget who it was. Sorry, but it's right. Once you start looking, it's
Brandon,
yeah, it was
I
that
forget the name.
yeah, it was that oh gosh, I always forget the name. the genus name. The Aurantia. Yeah,
it was Aurantia something with the species name.
Tulasnia?
Tulasnella.
Yes, that's right. Yeah, I think Brandon, you may have suggested it, or somebody did, but it's right. I mean, I looked at the microscopy and it's like, yeah, there's just these big long rows of conidia that some of them are truncate. And it's like, well, once you see all that, it's like, wow, I guess that's what it is. And so thanks. Yeah, that one. Oh, and Dorothy, are you still there, Dorothy?
She left.
Oh, I wanted to tell her. I went up in my yard and dug up the ground underneath where the marasmias were, and there was a live root of looked like a thin root of maybe some sort of wild shrub or something, because it's right near a border of an unmowed area. So they're probably rotula, but that's not, typical of Marasmius rotula to be growing from a live buried root. But I did the next day get out there and do that. So I didn't know what these were at first. I thought they might be some sort of Anacebaceae. So there's like 5 genera now that you have to worry about, which actually to me it makes the Anacebaceae a little bit easier than when they were all Anacebes, because now at least if you do a little bit of microscopy, you can often narrow it down to genus Anacebe, like I did with these 2 little guys. So these were under Scots pine, and I suspected they might be some sort of one of the genera in the family Anacebaceae. So how do I know it's an Anacebe? We don't know yet from the spores, these ellipsoid or slightly bean-shaped spores. We still don't really know because these look like Chylocystidia. It looks like a palisade of Chylocystidia. Now this other bowling pin shaped thing, that might also be one. And this looks like another just an area of the gill edge that's got a palisade of Chylocystidia. I think that's probably what's going on there. But now, when I got into other areas of the smash mount, I started finding these cystidia that had thicker walls. This one looks like it has thicker walls, and it was not a part of one of those palisades. So there were a lot of these other cystidia. So these are pretty likely Pleurocystidia. Once you find cystidia not right on the edge of the gill, and a lot of times the Pleurocystidia will dislodge in your mount if you do a smash mount, especially, and they'll just be floating around in the liquid. And so I'm saying these are a true Anacebe based on the presence of Pleurocystidia. So look at this one here. Look at the bottom part. Look how thick the wall is. You can see how thick the wall is there on the bottom part. So that would make that a metuloid if it's truly thick wall.
Dave, can I ask a question real quick? I agree.
Sure.
Based on this photo, they look like metuloids. But haven't for the last couple weeks, haven't we kind of come to the conclusion that for it to be an Ocypete proper, it needs to have nodulospores?
No. There's an if then there, but it doesn't go both ways. The converse is not true. If the spores are nodulose, then you probably have a true Ocypete.
Gotcha.
I don't know of any counterexamples to that. But if you have a
Okay.
true Ocypete, you don't necessarily have nodulose spores.
So there are some species
there's quite a
within
few.
true Ocypete.
There's so many true
Oh
Ocypete.
yeah.
And quite a few of them have these ellipsoid to slightly bean-shaped spores.
So, and just for all of our understanding, metuloids, chloro, and Pileocystidia.
Yeah, I didn't look at the Pileus here. A lot of times I'll just look at the gill. It's so
I'm sorry,
easy to.
I'm sorry, Pleurocystidia.
Pleurocystidia, right.
Pileocystidia.
Well,
Yeah.
so with these things, if you really want to work one of these things up, you look at the Pileus, you look at the surface of the stipe. I mean, these little things, like I wanted to preserve these also. So I just hacked off a little piece of gill. It's so easy. I just hacked off a little piece of gill. I think I laid out also I think I did lay the cap down for a half an hour or so, and I got a few spores. Sometimes you can just see a thin film on your microscope slide, and then you know you've got enough spores to scope. In fact, that's actually a good amount of spores to scope, because then you don't have that like too many of them that are just crowding each other out. But
And now that I've asked the question, and I appreciate the answer, I do remember you saying, if you see nodulospores, then you know you have an anosopy. Not necessarily anosopy have those nodulospores.
yes, I know.
Yeah.
I know of no counterexample to that conditional statement. If you see nodulospores, then you have an anosopy. I don't know of any counterexamples. Same thing with the pleural cystidia. And I did ask Jacob Polk about that. He spent some time with methane out in the field, and he learned a fair amount about anosopy. And he said he was pretty sure of the same thing. If you find pleural cystidia, then you know you have a true anosopy.
Cool. Thank you. The more that we talk about these, maybe I'll remember exactly how to ID them.
Well, these are hard to ID.
Oh,
I mean,
to
to get them to genus.
Yeah, to genus, right?
Yeah, there's a few that are not really that hard. Oh, well, a few of them that used to be not that hard, they have different names now. So, anyway, yep. I don't know what these guys are.
Thank you, and hang on to your pennies because we're not going to have them in a few years.
Oh, that's a good point. Let's go to Canada and get some pennies.
Why not?
Oh, what? They're going to stop making pennies.
I think they already stopped in Canada.
Yeah,
Oh, they already stopped in
They
Canada?
stopped in Canada already. Yeah.
Oh, so you have to go like to Europe, to get a penny.
Yeah, but
Well,
they'll
we
be
can
circulating around.
Yeah, I know. I'm just being facetious. There'll be pennies. Yeah, they'll still be around.
So they stopped actually minting them here in this country, right, as well.
They
I guess
made their
they did.
last shipment of blanks.
Oh, they're
So
going to
when that
stop
is used
soon.
up.
So the last dated year could possibly have some low mintage in at least one of the mints, because I think they're still using 3 mints: Philadelphia, Denver, San Francisco.
Mm-hmm. Yeah,
A low mintage.
Did you know that the outer shell is copper, but the insides are aluminum?
Oh, really? I didn't know that.
Yeah. Yeah. Don't get cheap on me. Yeah, they did get cheap on us. Yeah.
I wondered how they managed to make them, because there's more than a penny's worth of copper in a thing of this mass. So it's
It costs
aluminum
3
in
cents
there.
to make a penny.
Yeah. Yeah. I found out about this in high school a long time ago. Just stuck in my memory.
Might surprise you to learn that nickels are not made of nickel.
No, nickel. Nickel will be even more expensive than aluminum. So, yeah.
Okay, does anybody else have anything to share that they don't want to wait until next week? Okay. All right, well, thank you everyone for joining us, and I hope you enjoyed that chemical lecture in the beginning about fat fungus, white nose. And we'll see you all, hopefully most of you, next week. Have a good night.
Good night. Thank you.
At the 4A on Sunday.
Yeah, I think we're coming. We should be there.